Hello to everybody and welcome to our today webinar. My name is Mike Posner. I'm the Medical Director for the National Nutrition Institute in Europe, Middle East and North Africa. And it's my pleasure to share a symposium on this special day on the 17th of November, the World Prematurity Day. I think it's the day dedicated to babies born too soon and to their parents. And it's also my great pleasure that we have organized this symposium in collaboration with the EFCNI, the European Foundation of Neonatal Care, that have developed a nice, interesting project and is now working on the further distribution of these European standards of care, which aims to provide really standards to improve the care of the preterm babies on every infant. And here we share our vision that we really help the babies to be cared in the best way. And that is also the aim of the symposium. And I think this objective very much is represented with the content of the symposiums. What could be better and even more suitable than the World Prematurity Day to talk with experts in the field of nutrition about the situation and the progress in research of preterm babies. It's 10% of all babies, and I think it's worth to exchange in sometimes the new developments in this area. So I will now introduce one of the first speakers is Professor Nick Ambleton. He is a consultant neonatal pediatrician and professor of neonatal medicine, Newcastle University in the UK. He qualified with honors in medicine from Newcastle University in 1990. Doctoral thesis was in 2002 on the protein requirements in preterm infants. He is chair of the working group on donor human milk, British Association of Perinatal Medicine, and I think he's also at the ESPICA elected member of the Committee of Nutrition in 2014. Currently, Professor Ambleton is leading different research programs. It's about early feeding and preterm nutrition and the impact on supplemental lactoferrin, the preterm infant microbiome and metabolism, the growth, cognitive, metabolic outcome in children who were born preterm into late adolescence, the impact of exclusive human milk diet in very preterm infants, and qualitative studies exploring reproductive and/or neonatal losses. Professor Ambleton is an associated editor of pediatric research. He had published more than 200 peer-reviewed articles and has more than 7,000 citations. I welcome Professor Ambleton and I hand over to your topic.
Hi, my name is Nick Ambleton. I'm a neonatologist in the UK in Newcastle upon Tyne, and I'm going to be speaking in the next 30 minutes on 10 key concepts in neonatal nutrition. And these 10 concepts, first of all, the first one is that stores are limited and demand is high. Second, nutrition is more than nutrients. Third, we have one brain for life. We live in a microbial world. Necrosis enterocolitis is common and devastating. Not too little, not too much. Nutrition throughout the life course. Nutritional status. The importance of mother's own milk. And finally, teamwork. So I'm going to go through each of these 10 concepts quite quickly over the next 30 minutes or so. First of all, and most importantly, are that in preterm infants, stores are very limited and demand is incredibly high. Here's a picture from our neonatal unit that I took last year. And I think you can appreciate the complexity of neonatal care. And these are all the things that as neonatologists on the NICU we talk about every day, how much fluid to give the baby and CPAP, blood pressures and all these kind of things. But in that context, nutrition is often overlooked because NICU care is incredibly complex and tends to be focused on the cardiorespiratory management. And all the time we have monitors that will monitor cardiorespiratory status, but very few monitors that are really focusing on nutrition. So stores are limited and demand is high. Here's a picture of Pogacar, who's a Slovenian cyclist who just won the Tour de France. And this guy cycles up and down mountains for a living. And he consumes around about 100 kilocalories a kilo a day whilst he cycles up and down mountains. Put that in comparison to a preterm baby, where we know that energy requirements are around about 120 or 130 kilograms a kilo a day. So preterm infants are doing something that requires 20 to 30% more energy than the most demanding physical activity known to man. And importantly, around about 70% of all that energy expenditure is in the brain. If you want to consider protein requirements, this is how many Big Macs an adult would need to eat to be equivalent to protein requirements in preterm babies. So what are babies made of? We need to have some understanding perhaps of body composition in such a premature baby. And this is an amalgamation of work done now maybe more than 30 or 40 years ago. And what this shows you is that the mineral content of the body only weighs about 1 or 2%. Lipid accounts maybe only for 1 or 2% in babies born at 25 weeks, but closer to 15% at term. Protein is relatively constant, around about 10 or 12% of weight. But this means that most of the weight of the babies that we look after is actually water. So a 24-week baby is about 90% water by weight. This means that if you weigh 500 grams, there's only about 50 grams of solid matter. So here's a picture of Theo, a little baby that we looked after a couple of years ago, weighing only 350 grams. So he has to survive with less than 40 grams of lean tissue, yet somehow he's able to achieve this. So when we consider how much energy might be contained within that tissue, let's compare the fact that a preterm baby with 50 grams of non-hydrated tissue is about the same as a chocolate bar. When you put those energy requirements together, they're actually pretty similar. But the available energy left to this baby here is probably only 50 grams. And this means that a preterm infant is perhaps only 1 bite of a chocolate bar away from death due to malnutrition. Intakes really matter. So over the first week, this study showed that for every extra 10 kilocalories a kilo of energy you got, you had a 4.5-point advantage in developmental outcome at 18 months of age. And for every gram of protein, about an 8-point advantage in developmental outcome. So we need to think nutrition. We need to start nutrition as soon as the baby gets to the neonatal unit, both with buccal, colostrum and standard parental nutrition. We need good guidelines, audits, and we need, importantly, to avoid malnutrition. However, nutrition is much more than nutrients, and none of these nutrients function in isolation. So I like to recognize that nutrition is made up of lots of different components. Of course, we need to consider all the different nutrients, proteins, fats, micronutrients, but we also need to be aware of the functional components, such as human milk oligosaccharides in breast milk. We need to be aware of all the different microbial aspects that are induced by breast milk or the environment that we live in, and then all the technical or socio-behavioral or psychological aspects of nutrition. And all of these 4 components need to work together. So we need, as I say, to be aware of all the different aspects. We need to provide good leadership. We need to think carefully about the NICU environment and about functional components, but also be aware that nutrients don't function in isolation. Along the x-axis here, I've depicted protein intake, and up the y-axis outcome. This could be lean mass or developmental outcome. And what we see is that a good amount of energy intake, as the energy intake increases and protein intake increases, that outcome improves. However, if you don't provide enough energy, simply providing higher protein won't get you the benefit that you want, because the amino acids will simply be catabolized. So we need every nutrient to build a brain. This is what it looks like if you provide enough macronutrients and micronutrients. But this is what it would look like if you continue to provide enough macronutrients without the micronutrients. And those micronutrients might be phosphate, but equally they could be DHA or B12 or iron or these sorts of factors here. We only get given 1 brain for life, and this is really important in preterm babies. In most primates, brain growth is very rapid, particularly in the 2nd and 3rd trimesters, so that by term, most primates have a relatively mature brain. In comparison, the rapid period of brain growth in humans occurs towards the end of the 3rd trimester and in the first few months after term. So humans, in comparison to most primates, are born relatively immature. And between 24 weeks gestation and 2 years of age, humans acquire about 90% of their final adult volume. Now here's a picture of a rhinoceros with a very large head, but when you look at the brain is actually relatively small. In comparison, the brain of a human baby fills the entire cranial cavity. Here's another way of looking at it. Along the x-axis, I show you body weight as a logarithmic scale, and up the y-axis, brain weight. And you can see that most mammals plot out along that average line. But humans are off the scale at the top here. And in comparison, the human brain is around about 10 or 20 times as big as most other mammals. And everything about placental function and breast milk composition is designed for us to grow a big brain. That is the whole focus of nutrition in early life. Now, not only do we acquire a lot of brain volume in this process, in this time period, but there's obviously lots of different processes going on. Neuronal migration, particularly active throughout second and third trimesters, apoptosis, synaptogenesis that may not complete until a few years of age, and myelination that may even continue into adulthood. And all of these processes are under the control of growth factors, particularly IGF-1. And now there is accumulating evidence that the amount of nutrients we give babies, particularly macronutrients, influence IGF-1 levels. So there is a direct link between nutrient input now and control of various neurological processes. And brain development, of course, is very complex. There are lots of different anatomic regions and lots of unique developmental processes. And all of these need to take place in a time-coordinated sort of pattern for us to bring and develop a normal brain. And so these complex circuits that need to be formed all happen to happen in sequence. And if you don't do the first few steps correctly, you won't be able to do the subsequent steps appropriately either. So the impact of nutrient deficiency on the brain depends on the amount of that nutrient, the type and timing of nutrient deficit, and the duration. So here are 3 theoretical examples. So A might be an example of macronutrient deficiency, which is particularly common in the first few days. B might be an example of a nutrient like iron, which perhaps you don't need lots of in the first few days, but after that becomes important. Whereas nutrient C might be something really important throughout the third trimester and infancy, like choline or DHA. And so a lack of any of these nutrients will have a different impact because the insult is occurring at a different stage of brain development. And there are lots of mechanisms that might link nutrients to brain development. Of course, we need all the micronutrients for tissue substrate. We then need energy to drive the system, particularly carbohydrates and lipids. We need lots of signaling and growth factors. So, for example, sphingomyelin and phospholipid that you find in the milk fat globule membrane, but also lots of growth factors. Next, we recognize that there are lots of nutrients that impact on gene expression. This is particularly true for folate, B12, and iron. We know that breast milk reduces neck and sepsis, and neck and sepsis are particularly damaging to the brain. And then finally, all the microbial aspects, such as the role of prebiotics like human milk oligosaccharides, lactoferrin, and perhaps probiotics. So we only have one brain for life. The practical relevance of that is that we should try wherever we can to prevent neck. We need to maximize use of mother's own milk. We need to meet macro and micronutrients as soon as possible, and we need to regularly review nutrients. Next, we need to recognize that we live in a microbial world, and the gut really is the inner tube of life. Hippocrates says that all diseases begin in the gut, and when we think about preterm babies, that really might be true. 80% of all the antibody-producing cells in the body are located in the gastrointestinal tract, which means that the gut is the most important part of the immune system. There are more microbial cells in us than human cells, and many, many more microbial genes than human genes. So really, we might think of humans as a superorganism involved in a lot of metabolic and immune processes, but it's important to recognize that human development would be impossible without these microbes. And when you add up the weight of all these microbes, they come to about 1.5 kilos in an adult. So we've evolved in a microbial world, and there's this continual tug of war between how we manage beneficial microbes, but also how we exclude harmful microbes. And that challenge is particularly acute for a newborn baby who has to learn on the one hand to establish tolerance, whilst on the other activating immunity against pathogens. And you might ask yourself the question, how on earth does a 24-week baby who is supposed to still be in utero understand which are the healthy bacteria and what's supposed to be a pathogen. We know that the pattern of bacteria change over the life course. They're very different in adults to what they are in babies when they're first born. But we have very little understanding about what the healthy pattern should be in a preterm baby, because of course preterm babies were not designed to be here in evolutionary terms. When term babies are born, of course, they come into contact with mother's secretions, skin, and these sorts of things, and then they go home and get into contact with all the things in the environment. And we know that initially the bowel is colonized by anaerobic species, particularly bifidobacteria and Bacteroidetes, which create and sustain a low oxygen environment. And into this mix, breast milk and breastfeeding support successive waves of colonization. But just think how different this is for a preterm baby. Over the first few seconds, minutes, hours, and days, the life and environment of a preterm baby is very different to that of a term baby. Furthermore, they're touched by hands that are washed with soap and antiseptics. We put bits of plastic into them, we give them lots of antibiotics and maybe H2 blockers. And then even when we give them breast milk, we have to give it in abnormal ways that involve coming into contact with plastics and various aspects of processing. So it's perhaps not surprising that microbial aspects of preterm babies are very different from that in term babies. And to some extent, all preterm infants are dysbiotic with a lack of diversity and absence of key species like bifidobacteria. And we're particularly interested in many of the metabolic products of these bacteria rather than simply these bacteria. Now, this is really important because, of course, bacteria have a key role in the development of NEC and sepsis, and we need to recognize, and I think this isn't widely appreciated, that children are more likely to die from necrotizing enterocolitis or sepsis as a preterm baby than all cases of childhood cancer combined. And so going on in the gut is this continual interaction between microbes, nutrients, and the immune system that changes over the life course. And we can think of NEC perhaps as a breakdown in an interaction between these three different elements, but we don't really understand what might be going on. So NEC is a really terrible disease. It's a pathological term applied to a clinical syndrome. We know it's multifactorial, and unfortunately there's no single diagnostic test. It strikes without warning. Around about half the babies who develop serious NEC require surgery, and about half of those who require surgery die. So this is a really serious disease that tends to dominate a lot of the feeding practices on a neonatal unit. And NEC is probably the end result of lots of different pathologies, and it presents in lots of different ways. And perhaps one way of viewing NEC is that it is one end of a continuum of gut health. So whilst NEC is the visible tip of the iceberg below the surface, going on below the surface are all these other important aspects of dysbiosis, inflammation, and metabolic and epithelial health. So we like to think of NEC perhaps as the tip of the iceberg of gut health, with lots of other factors lurking below the surface, such as the role of inflammation, feeding, and growth. So within the gut lumen is this continual interaction between microbes and nutrients. Depending on the type of nutrients, we change the pattern of gut microbes. But in turn, the pattern of gut microbes influence the pattern of nutrients and metabolites that we might see in the gut. Whilst providing adequate nutrition is really important, it's also important not to provide too much. Generally, I think with preterm babies, we don't need to be too concerned about making them obese. We really need to focus on providing with enough nutrients to grow a good brain and match lean mass accretion. But during acute illness particularly, it's important that we don't overload the metabolic machinery. And this has been conceptualized by Cohen-Houston and others into these three phases of acute illness. The acute phase, when ventilation and inotropic support is increasing. The stable phase, when you still need inotropes or ventilation, but perhaps the amount of support you provide is decreasing. And then the recovery phase, with the vital support of organs dropping down. And if we think about what's going on in those phases, we know that inflammation is peaking during the acute phase, but this is associated with lots of metabolic changes. So endogenous production of glucose goes up, and there's a suppression of tissue synthesis. And these various clinical processes are matched by changes in different levels of hormones. And so we see an increase in growth hormone and thyroxine during the acute phase, but we also see a suppression of growth factors like IGF-1. This is true in term babies and children. And in preterm babies, IGF-1 is probably suppressed even more. So during stress and sepsis, the body tries to look after itself by producing a surge of catecholamines and steroids. And this in turn increases endogenous glucose production, increases peripheral insulin resistance, which provides more glucose for organs. But also we start breaking down proteins and fats, and this releases a lot of amino acids and free fatty acids. And so during this stage, it's very important that we don't overload the system by providing lots of parental nutrition. Another really important concept is to appreciate the importance of nutrition throughout the life course. We know, of course, that what happens to you as a baby is likely to influence what happens in childhood. But now there's accumulating evidence that how you grow as a baby, but also as a fetus, maybe even before that, these exposures influence how you turn out as an adult. Now, of course, our genome is fixed, but exposures during critical periods in early life, during sensitive windows, impact on later health and disease. And this whole area has now been conceptualized under the developmental origins of health and disease, with work going back to McCants in his rat studies in the 60s, but developed particularly by David Barker in the 80s and Alan Lucas in the 90s. And various terms have been proposed, but these are now conceptualized within this overall term, developmental origins of health and disease. And effectively what this does is recognize that exposures perhaps even before conception or in utero or in the postnatal period have an impact on later life. And there are likely to be lots of different mechanisms, both epigenetic exposures, but also toxic exposures and environmental factors, interruptions in preterm babies of the normal pattern of brain development, but also, of course, importantly for us, lots of different nutritional and microbiomic exposures. So this has real relevance to management of preterm babies. There are lots of factors that might associate preterm birth with later outcomes. And we know that in term babies, fast or slow growth perhaps may result in different later outcomes. But unfortunately, in preterm babies, these differences in growth, it's not clear to us in later life whether these are due to differences in growth or actually differences in dietary nutrients. So whilst a lot of people are concerned about this in term babies, I don't think we can consider that in preterm babies that nutrients are necessarily going to cause harm. We do need, as I say, to focus on the importance of catch-up growth in babies who haven't grown properly, providing sufficient nutrients for both the brain and lean mass secretion. And it's really too simplistic to say that rapid growth is always bad for you. So we definitely need follow-up of large trials. And the overall factor that I think we can all agree on is that giving lots of mum's breast milk will always be good. How do we assess nutrition in preterm babies? Well, nutritional status is really quite a complex concept. If you ask people to assess nutritional status, then people look a bit confused. It's easy for us to understand how you might assess respiratory status. People would look at respiratory rate or cyanosis or check a blood gas. And similarly, if you ask people to assess cardiovascular status, they would look at the heart rate and capillary refill, oxygen requirements, these things. But when it comes to nutritional status, people are a bit confused. What is it that we should be measuring? And most people, of course, recognize the importance of monitoring growth and using a growth chart. And people like to think that there might be some blood tests that would help us here. Unfortunately, most blood tests in preterm infants are fairly unhelpful. You can take a blood sample and send it off to the lab and ask for nutritional status. Unfortunately, you don't get a sort of report like this. You get a lot of biochemistry back. And very little of this really tells you how you're doing in terms of providing nutrition. So we need to consider this life course approach to nutritional status. So what happened in the past determines your metabolic capacity to handle nutrients in the present time. And that in turn impacts on later risk of disease, particularly things like diabetes. And there are lots of different aspects to this. And as I said before, you could think about all the past exposures, which might be past exposures to the mother or to the fetus. What's happening in the present time, your ability to handle glucose or lipid, or what your body composition is like, or what diseases are ongoing. And these in turn impact the risk of later diseases, which may be metabolic diseases, but also things like brain outcome. So this is a really complex concept to get your head around sometime. And so we've reduced it to the ABCD approach for babies on the unit. So first of all, A, consider anthropometry. B, think about bloods and biochemistry, so particularly sodium, phosphate, calcium. C, think about the clinical factors, whether the baby is very wasted, whether he has rashes, and what specific clinical factors are there, such as a stoma or lung disease. D is to assess the actual dietary intakes, particularly of your micronutrients, for example, exactly what you got over the last 24 hours or so. And then to consider the wider aspects of nutrition in the environment, so particularly what resources you have, whether you're supporting the mother to provide breast milk and feed readiness and these sorts of things, and particularly emphasizing the importance of involving and explaining all of this to the mother. Now, mother's own breast milk is almost always best in every preterm infant. And numerous studies have shown this is really very important. We've learned a lot in the last 10 or 20 years. But prior to this, I and perhaps many other people used to think of milk just as a fluid that you tipped into this great big long tube, that you suck out the fluid and nutrients, and at the end you produce waste product. That was how many of us thought about milk 10 or 20 years ago. But we've learned that things really are much more complicated like that. And we can think of breast milk almost as if it's a biochemical fluid and a means by which the mother can signal to the baby. And there are all these various aspects, such as the metabolites produced in the gut or interaction with the immune system or microbes, that are really key to understanding nutrition and gut health in preterm babies. So as I've said before, we need to think of nutrition as being more than simply the nutrients you give babies, and in turn, therefore, think about breast milk as being more than simply a food. And there are many, many examples of why breast milk is better for preterm babies. Just about every key morbidity you can think of in preterm babies, be it neck, sepsis, retinopathy, or lung disease, is reduced by exposure to mother's milk. We know that babies who get good amounts of mother's milk have better metabolism in later life, so either lipid profiles or bone mineral density. And also we see better brain development in babies exposed to lots of their mother's milk. And there are really numerous studies that support this idea now. So here's one study conducted in France in a large cohort. Up the y-axis, there's a measure of brain developmental outcome. And then there are 4 groups here: babies who didn't get any mother's milk, babies who just got mother's milk in the NICU, mother's milk for 2 months after NICU discharge, and then milk for longer than 2 months after discharge. And you can see this quite nice linear relationship between exposure to mother's milk and better brain outcomes. So I've rushed through those 10 really important concepts. I guess we could come up with a few more. So just to conclude, stores are very limited and demand is high in preterm babies. Nutrition is much more than nutrients. We need to think about all the functional components, microbial components, then all the behavioral aspects of feeding. Babies only have one brain for life. We really need to protect and grow it as well as we can. We live in a microbial world, and this in turn influences our risk of neck and sepsis. Whilst we need to make sure we give babies plenty of nutrients to grow properly, we need to avoid overloading the system during acute illness. And nutrition is important throughout the life course. How we look after babies now may influence their risk of disease when they're much older. Nutritional status is quite a complex concept, but as I say, I think you can use the ABCDE approach. And there's an overwhelming amount of evidence showing that mother's milk is always best. And finally then, teamwork makes the dream work. What do I mean by this? Well, we need to appreciate that it's not just doctors or not just nurses or dietitians involved in providing good nutrition to babies. So say we have now got good data to show that there is a dose response reduction in just about every neonatal morbidity for human milk, and therefore we're obliged to make sure that we educate staff, hospital managers, children, society, and parents about the importance of this. And we have to do that by adopting a holistic approach, by demonstrating good leadership and advocacy, and providing empathic, careful support for mothers who are deeply stressed at this period in time. So we clearly need a lot more work, but getting good teams together is key to improving outcomes through nutrition. So why is it that mother's own breast milk provides all these advantages? Well, here's a list of about 200 different components that we know are present in human milk, that currently most of these are not present, or at least they're not present in significant quantities in formula milk. And it's going to be impossible to recreate this mixture this really complex interaction between multiple components. This will never be replicated in formula milk. So formula milk might incrementally change the composition, but we will never ever be able to replace this complexity of human milk. And there in the middle is Harrison, who is a 23-week baby, who I've kept in contact with, learning at around 39, 40 weeks corrected age to breastfeed from a mother who very patiently, for months and months of neonatal care, continued to provide him with her own breast milk. So I'm going to finish there, just again to emphasize that the work we do is always part of a team effort, whether it's clinical work or academic work. Here's another cycling analogy. Here are the teams that win the Tour de France things, and they only win it not because the guy in the yellow is the best, but because the whole team worked together to get the yellow guy over the finish line. And so really just to emphasize that nutrition is everybody's responsibility, and that it involves parents and nurses, dietitians, but a whole range of therapists and pharmacists, family support workers looking after the psychological well-being of the mothers, the families, lactation support, etc. So providing good nutrition, key to long-term improvements in health for our preterm babies. So if you want to get in contact with us, our website is there, neonatalresearch.net. You can contact us from there or look at any of our slide presentations there again. And I'll be delighted to take any questions. Thank you for your time and attention.
It's now my great pleasure to introduce to our second topic and a very renewed speaker, Professor Hans van Kodo. He is the division chair of Emma Children's Hospital Amsterdam, professor of pediatrics at the University of Amsterdam and the Free University, the member of the National Health Council appointed by His Royal Majesty, founder and director of the Dutch National Donor Milk Bank. He received his medical and pediatric training at the Erasmus University, Rotterdam, and performed research for his PhD on nitrogen metabolism in preterm infants we are all getting a little bit older, in 1993. Postdoc intestinal amino acid metabolism in a piglet model revealed novel insights into nutritional management of the preterm infants in the lab of the late Peter Reed's Clinical Nutrition Research Center. He was in Texas Children, Baylor College of Medicine, Houston. In 2000, he was trained as neonatologist in Rotterdam. He is a professor and chair of neonatology in 2004. I think he's well known. He served in many international councils, including European Society of Pediatric Research, International Pediatric Research Foundation, the European Society of Pediatric Gastroenterology and Hepatology and Nutrition, and he served as chair of the Committee of Nutrition. Approximately 350 peer-reviewed articles predominantly on neonatal nutrition and metabolism. He's associate editor of several nutrition and metabolism journals, and I think he supervised over 40 medical doctors, PhD students. Please, Hans, go ahead with your lecture.
Good afternoon. Thank you very much for joining this webinar. It's a special day today, so I'm glad we can discuss issues that are related to infants born preterm. And I was asked to address the topic human milk banking. I have nothing to disclose. I will discuss with you some really exciting results with regard to a study that we are currently performing, which is published in medRxiv earlier this year. That was supported by the Foundation to Support the Emma Children's Hospital. What our learning objectives are for today is that I will talk about the benefits of human milk for preterm infants. I will discuss mother's own milk versus processed donor milk, give you the latest ESPRAM recommendations, and the last part of the talk will be on this new developments of COVID and the use of donor milk. We all know that breastfeeding for term infants has beneficial effects on the long term. Blood pressure is reduced, cholesterol levels are lower, your risk of diabetes or overweight is much lower than when you're formula fed, and also you gain some around 2 to 3 IQ points. So all beneficial effects of breastfeeding. And also for preterm infants, some of those results are there as well in the literature. This, for instance, is from Acute, a paper published already 20 years ago on blood pressure measurements of formerly preterm born infants, now at the age of 13 to 16 years. And what you easily can see from this slide is that, for instance, infants who are fed human milk had a diastolic blood pressure of 62 millimeters of mercury, whereas those who were fed preterm formula had a diastolic blood pressure of 65 millimeters of mercury. A similar difference was found for the mean blood pressure. And one can easily say, well, this is not really a huge difference, so who cares? But it does. These results correspond to a reduced risk of coronary heart disease of around 10 to 20% on the long term, and also a reduction in stroke risk of about 20 to 30%. So these small differences are really important for the long term. But not only blood pressure has been studied, also IQ, and where I just said that for term infants, the difference in IQ is about 2 to 3 points. For preterm infants, it's much higher. It's around 10 IQ points difference at 8 years of life. Again, shown by the same group, published now 30 years ago. Of course, nowadays, these are old studies, and nowadays you want to have these wonderful new data, and this Cochrane library usually presents those data in a very elaborate way that everyone knows, and that also the quality of those kind of reviews is very good. So I was very much surprised and was happy that in 2019, there appeared to be a review on formula versus maternal breast milk for feeding preterm or low birth weight infants. And they said, well, we are going to use randomized controlled trials comparing feeding with formula or human milk in those infants. But these were the results. There were no eligible trials were identified. And that, of course, has to do that one just cannot do any randomized controlled trials for own mother's milk versus formula, because the mothers who want to provide their children with own mother's milk do not want their child to be randomized to formula, or the other way around is just not possible. And then for all kinds of other issues, there are problems interpreting other regular research projects that have more just looking at different populations. Because, for instance, if mothers are very ill, it might hamper breast milk provision. And the disease of the mother may affect the outcome of the children. Maternal stress in the most sick babies might reduce breast milk provision to just those babies. We also know that higher IQ, higher socioeconomical status, or non-smoking are associated with more breastfeeding and therefore better infant health. So that's on the other side. And then there might be residual confounding, just confounders that we are not yet aware of. So usually we only talk about associations, and not only the associations from studies from 20 or 30 years ago, but for instance here, Belfort et al. Published a paper in 2016, a cohort study, so all those studies are cohort studies, on 180 preterms, less than 30 weeks of gestation, and they were looking at the correlation between the numbers of days on which own mother's milk intake was more than 50% of the total enteral intake during the first 4 weeks of life. And what they found was really, really important. They found that every day that there was a provision of more than 50% of all mother's milk was associated with a half IQ point at age of 7 years. So that is really a remarkable result, but we have to say it's only an association. Another study from the glutamine trial from the US with almost 800 extremely low birth weight infants who were seen 2.5 years later at follow-up rate of 83%. They found that here in red, the infants were fed only formula. In green, they received more than 80% of own mother's milk. And in between the colors are, as depicted here below, were different percentages of the total intake of mother's own milk for those infants. And one can clearly see that the higher intake of own mother's milk was associated with a faster approach, a faster speed to reach full enteral feeding. The length of stay was reduced when the infants were fed more own mother's milk. Rehospitalization rates were reduced, and the Bailey 2 mental developmental index score was increased when the own mother's milk intake was higher. Again, these are just associations. Another study, a meta-analysis of cohort studies, looking at own mother's milk versus preterm formula, but then as a result, the result was retinopathy of prematurity. And here from this forest plot, one can easily see that again, there is an association that favors human milk in the reduction of the incidence of retinopathy of prematurity. Again, associations. Seeing all these associations, I was very curious what was happening in our own unit, and that was also about 10 years ago. And we looked at the first 10 days of life. And what you can see here from these pie charts is that in the beginning of the first 10 days of life in our unit, the majority of the infants received formula. And then at day 5, it was about half of them received mother's own milk. And at day 10, it was more like 80% or so. So especially the first 10 days, infants received predominantly formula. We looked again in a cohort on outcomes during the first 2 months of life in those infants. We looked at 350 very low birth weight infants, and we exactly determined the intake of mother's own milk or preterm formula during those first 10 days. And then we looked at the event-free survival, so sepsis or NEC, so infants that survived without sepsis or necrotizing enterocolitis. And in the first 5 days, here depicted on the left-hand side of your slide, you can see that the event-free survival was highest in infants who received some mother's own milk. And there was not really a dose-response relationship. On the day 6 to 10, there seemed to be a dose response, indicating that the higher amount of days that received more than 50% of own mother's milk, the more infants survived for 2 months without having any sepsis or necrotizing enterocolitis. Again, these are associations. But seeing the low number of infants starting on mother's own milk directly following birth, we decided to install a human milk bank here in Amsterdam, and that was founded 10 years ago. It's not a very new thing actually, because here alongside the canals here in Amsterdam, there's a house that has this stature in the wall that says that in 1662, there was already, one was already talking about wet nurses. So rich mothers or rich women who gave birth to children did not want to provide breastfeeding to their own child, but they hired other women that just gave birth to their children and to provide breastfeeding to their own children. So that was 1662. Directly following the Second World War, there was a Dutch human milk service that ran from 1984 till 1973, because at 1973, people thought, and it was such a good marketing, people thought that formula was as good or even better than own mother's milk. But it's fun to see how the milk was collected and freeze-dried and then given to weak newborns born in those years. Nowadays, it's completely different. We have now almost 250 human milk banks that are active in Europe, and you can see this on this screen. And there are 20 more planned, as being shown by the EMBA, the European Milk Banking Association. What do people do at human milk banks? Well, the milk is delivered. In most countries, the women that provide the milk, that have thus a natural surplus of milk after giving their own child, of course, also breast milk, they deliver it at the bank. They are not reimbursed for that, except for some countries in Europe. So they do it for altruism. It's checked, the temperature, it's labeled, it's stored in the freezer, then it's defrosted within 3 months after pumping milk. It's cultured to make sure that there are not a huge load of bacteria. Then it's pasteurized, which eliminates CMV and the bacteria. It's cultured again. It's analyzed, how much protein, how much energy is in there. It's freezed again. And then in our country, from Amsterdam onwards, it's transported to 5 NICUs, to about half of the country. And there it can be used within 9 months following pasteurization. And many people come and visit the milk bank, but it's just a few freezers next to each other. We pool one donor. We have a track and trace system, so we know exactly from which mother, which milk is delivered to which child. So that is always possible to trace back. So we know exactly who gets what. We pasteurize the milk. And like the vast majority of the milk banks around the world, we use Holder pasteurization. And Holder pasteurization is a safe method. We have been using it for years, and everyone has been using it for years. And you heat up the milk for 30 minutes to 62.5 degrees of centigrade, and then you cool it down again and freeze it again. This heat processing has an effect. Basically on all cells that are all living cells. So that's why it's killing all the bacteria and the viruses, but it also destroys all enzymes, all lymphocytes in there. It abolishes basically IgM. It has also a huge effect on lactoferrin, for instance, on several growth factors. So although it's a very robust way, it also damages the human milk. There are alternatives now being investigated in several research institutes, including a combination of the Wageningen University and Amsterdam University, where we look at all kinds of outcomes using high temperature short time, so that's for 72 degrees, but only for a few seconds, UVC irradiation, high pressure pasteurization, ultrasonification. There are a few more methods that are now being tested to see which ones are the best. If we look at And the randomized controlled trials, and now also looking at the Cochrane database, when comparing donor human milk versus formula, that is possible. And there you see some striking differences. Basically, by pasteurizing the milk and processing the milk, one can easily see that weight gain and also length and head circumference gains are reduced in infants fed donor milk as compared to formula. So weight gain, you can see here in the lower, is around 2.5 grams per kilo per day, less in infants who receive donor human milk. So that needs to be fortified if one would be favor of having a rapid weight gain rate. However, there are also some other benefits for the use of donor milk versus formula, and that has to do with the risk for necrotizing enterocolitis. You all know necrotizing enterocolitis is a devastating disease, and donor human milk has shown the capacity to reduce the risk for necrotizing enterocolitis. The risk ratio is 1.9, so there is a substantial reduction in it. However, the number of infants that are in those studies is relatively low. There are also some industry-sponsored studies included that may have a benefit from using the donor milk. And there are some studies that are performed in the 80s where the policy in the neonatal intensive care units is clearly different from what it is nowadays. However, if we look at the Escrow advice on donor human milk and preterm infants, we stated already in 2013, and still valid, that own mother's milk or mother's own milk is the first choice, and strong effort should be made to promote lactation. And when mother's own milk is not available, donor human milk is the recommended alternative. When neither own mother's milk or donor milk is available, then one should use preterm formula. Now I switch gears a bit to some really intriguing new data on COVID-19 and the use of donor milk. This is a huge effort of many different labs. Here you see them listed all. And we started this study at the beginning of the pandemic in April this year. Here you see all the people that are involved. So it's really a lot of people, both from Amsterdam, but also from the blood bank or the University of Utrecht, from the Wageningen University, and from a small company called Viral Clinics that has the capability of hosting the real SARS-CoV-2 virus, and we can do all kinds of studies with it. I'm going to start with showing you a small movie clip. It takes about 1.5 minutes that shows the possibility of using breast milk as a bridge till we have a vaccine.
In a world where millions of lives are under threat due to COVID-19, it is of vital importance to gain a better understanding of how the virus actually works in search of a cure. The surface of the virus particle is covered with approximately 100 spike proteins. They always come in groups of 3, in which the proteins are intertwined. Each individual protein consists of 2 parts: a globular head called S1 and a stalk-like structure called S2. One of the 3 heads is slightly bent, enabling it to connect to the so-called ACE2 receptor on a human cell. Once the connection between the virus and the receptor has been established, the human body activates the TMPRSS2 enzyme. This is a protein-cutting enzyme that proceeds to cut off the head of the spike protein. This causes a change in the structure of the virus. The S2 protein now grows longer, penetrating the human cell. The protein then retracts again, bringing the membranes of the virus and the cell very close to one another. At this point, the virus can enter the cell through a process of endocytosis. It engulfs the cell with its membrane. The virus has successfully invaded the cell and can now start to multiply, ready to overrun the system. However, we have strong defense mechanisms that try to prevent this from happening at all costs. Scientists recently discovered how our antibodies react. It's actually a very simple process. Our antibodies bind to the spike head, or S1, thereby preventing it from connecting with the ACE2 receptor and stopping the virus from spreading. Human breast milk from mothers who have had COVID-19 disease contains those antibodies. Breast milk may thus serve as a prevention strategy in this global public health crisis.
So this movie that was made available for us by Sensu actually already hinted towards the idea that we had that breast milk contains antibodies against SARS-CoV-2 virus, as we know that it already contains antibodies against many different viruses. Here I've listed them on the left-hand side, all kinds of viruses that we know breast milk has antibodies against. And for some of those, we also know that those antibodies are really proven to neutralize the virus. So herpes simplex, CMV, echovirus, Coxsackie virus, poliovirus, we already know by experiments that the antibodies in breast milk can neutralize that virus. So our question was, does breast milk already also contains antibodies against SARS-CoV-2? So we had lactating women participating in a study who had a natural surplus of milk, and a group of them were confirmed COVID-19 positive. Then there was a group that had a person in the household that had a confirmed infections, and the lactating woman developed COVID-19 symptoms, and then we had, of course, a control group. And here you see the characteristics of those groups. 3 of the donors who had conferred COVID-19 were also actually admitted to the hospital and got oxygen treatment out of the 29. 9 of them were suspected. They were all term delivering women with time between the start of the clinical symptoms of around, on a medium, about 6 weeks from onset till when we were collecting the milk and blood samples. And here on this graph, you can see that more specifically. Here on the left-hand side, start of the symptoms, and then from about 2 weeks onwards until 13 weeks, we were able to have cross-sectional milk samples from those mothers. We looked at with various ELISA assays to different parts of the virus to find antibodies, reactive antibodies. So we looked at the whole spike protein, but also the receptor binding domain, here depicted in green. We looked for IgG-specific antibodies, for IgA-specific antibodies, for total antibodies, and we also looked to the antibodies against the nucleocapsid protein, which is, of course, in the inner side of the virus. And here you see the results, and I will explain it. Here on the x-axis, you see the mothers the samples who are numbered. So the first 3 are the hospitalized women who donated milk, and then we from 4 till 29 are the ones that were positive on PCR. Then we have the 9 suspected, and then we have the controls. Here you see on the y-axis, first the serum. We looked at all the different antibodies, spike, IgG, receptor binding IgE, receptor binding total antibodies, and nucleocapsid protein antibodies. And here on the upper part, we looked at the milk samples, spike, IgA, and receptor binding domains. And what can easily be depicted is that for the confirmed PCR, we see lots of reactions. The higher the levels of the antibodies, the more yellow bright they are. And if they're negative, they are dark blue. So we found that about 80% of the proven cases, we found antibodies in breast milk, and in about 2/3 of the suspected, and hardly any on the controls. So that was important to find out, because now we thought, well, we might have a method to prevent SARS-CoV-2 infections for newborns. However, to provide breast milk to other individuals, one needs to be sure that it's a safe product. And we all know that some of the viruses can be transmitted through breastfeeding. CMV we know, HIV we know, but for instance, hepatitis B or C, we know that there's no transmission. So we wanted to be sure that it's not the case. And we were helped by several reports in the literature, and this was just published a week or two ago from Chambers et al. From the US. Here we see samples from women who had SARS-CoV-2. The squares who have a dark color are women who had symptoms, and the squares who are open, the women did not have any symptoms. And they looked at before the samples, before the mothers had a positive test result, and after they had a positive test result. And basically what they found, and also in other studies, that 10 samples of breast milk contained RNA, so that is PCR, but there's no virus cultured in the milk at any of the studies that are available now. So the question is whether antibody-rich breast milk can be used as preventive strategy. Just to be absolutely sure, one needs to have this safe product. So one needs to pasteurize the milk to be sure. And we compared Holder pasteurization to another alternative, which is called high pressure pasteurization, high pressure processing. And we looked at that machine. It's used, for instance, for fruit juices pasteurization, or for cheese, providing a very high pressure for only 5 minutes. We looked at the ELISA levels, and we saw that whether you looked at spike IgA against SARS-CoV-2, there was no effect on the high pressure pasteurization or the high temperature processing. But when we looked at the specific function of the secretory IgA, which is the dominant immunoglobulin in the milk, and we looked at the FAP region, which is this part, we found some striking results showing a difference between raw milk, here put at 100%, high pressure, which dropped to about 85%, and Holder pasteurization, which dropped to about 60%. And when we looked at the efficacy of neutralizing the virus, we used a specific, very robust system where we incubated for 4 to 6 days a fixed amount of a clinical isolate of the SARS-CoV-2 virus with a serial dilution of the milk and the blood samples. And we were just looking at the cell viability marker to see how effective either the serum or the milk was in neutralizing the virus. And here we see the same figure as I just seen before, but now in these upper part here in black, the neutralization, one can see that the serum was able to neutralize the virus in about 13% of the samples, whereas the raw and the high pressure pasteurized milk was able to do that in 26% of the milk samples. Holder pasteurization, on the other hand, lost all its potential to neutralize the virus. So Holder pasteurization was too robust, basically, to keep the neutralization capacity of the milk that contained antibodies of mothers who had COVID-19. So in conclusion, and I'm ending now, just 30 minutes after I started, breast milk of mothers who were previously infected with SARS-CoV-2 contains significant amounts of IgA against SARS-CoV-2, and that lasts at least for 13 weeks. Those milk samples have neutralizing capacity, also after pasteurization using a high pressure processing, but not when you treat the milk with heat. We think, and we're in the process of getting there, that breast milk containing antibodies can be used as preventive strategy to reduce viral load, not to cure disease, but to prevent it occurring by reducing the viral load and therefore the subsequent development of COVID-19. It looks very logical to provide that to all kinds of premature infants, to newborns, but one can also think of other vulnerable populations such as elderly. I thank you very much for your attention and I'm happy to address any questions you have. Thank you.